My pleasure to introduce my colleagues, Professor LEON GUO, and usually called by Leon. And he is my colleague as College of Public Health as well. He is a professor in College of Medicine. He is very famous for his hard work on this U-turn study. And also recently, in the past 2 decades, involved with a lot of child's respiratory diseases related to the environmental factors. He's now loaned from us to National Health Research Institute, which is NIH in Taiwan, and we count on him to transform the NHI to be more environmental oriented. So with this, he's going to share with you some of his research and policies that he has done in the past. Leon, yes.
Thank you very much for Dean Chen for the very nice introduction. Just a few words about the NHRI. So NHRI is about 1 hour and 15 minutes by driving, if not too much traffic. This is the structure. And these are some of the eggs, but it's called star clusters. I still don't know what to do with them. This thing is called living and resting. In Chinese, 生息. I had hard time finding out which one is living, which one is resting. And this one on the right side is having another red wine, which I think responded to a very famous song by Little Dan, a very famous Taiwanese singer, probably as famous as Deng Xiaoping. And many times we have visitors, and you can see some of our friends in front of the statue of the having another red wine. I think it becomes a fashion since this morning that speakers speak in 3 points. So I use 3 points approach. I had to change my slides very. What is special about Asia in environmental epidemiology? What are new knowledge demands concerning health of the next, or future, I should say, future generations? Is it possible to prevent environmental events? So these are the topics relevant to us. So what's special about Asia? We all know that some of the environmental problems are most prominent in Asia. So these are the deaths attributable to ambient PM pollutants in 2015, as nicely reviewed by the HEI study group. And you can see that the reddish portion, therefore the most significant hazards been in Asia. And why is it important for environmental epidemiology? This is the WHO Air Quality Guideline 2005 on PM10. I was trying to find the later version, but there is an update, but not a full guideline. And so I use this one. You can see that many studies actually were done by European and American studies, and also Australian studies. However, their exposure level have been rather low. What happened to the Asia cities and people they actually are exposed to a much higher concentration of PM10. And it's very similar situation in the PM2.5. So recently we did a study with some of the review, which got accepted, but I think it could be related to the review. But we can see that many European and American studies show lower level. This is 5 to 95% of the exposure level of PM10. PM10, sorry. And you don't see much effects in here. Why it reached more than 30, and our study was exposure median is 58.9. We found lung function effects. So the problem is, do countries from Asia need such data to convince their government to lower their exposure level? I greatly admire our Korean colleagues. This is a Korean station about a real-time air pollution report. But they essentially cover everywhere. They cover the whole world. So you will see that a lot of Asian red and orange and yellow, but more green on the other part of the world. And that's a summary where we should work harder, but we also need to provide environmental epidemiology evidence. The other thing is important factor that Brenda and many other researchers here warn us that health related to climate change is an important question. Only a small, I won't get into the depth, but the daily damage caused by climate change, Southeast Asia region is very susceptible. And there is certainly estimated cooling demands in up to 2050 is such trend continue. And you can see very red regions in Asia. So I think that's why environmental epidemiology is important in Asia. My second topic is what are new knowledge demands concerning health of the next generation. I will just talk a little bit, but changing living environment, including urbanization, man-made environment, and climate change. New exposures, observed effects caused by perinatal exposure and transgenerational mechanisms, which were unknown to us 1 decade ago. New exposures, these are starting from old exposures. PCBs were used starting 1920, started to be found to have health effects 30 years later. And then there were events, and then banned. You can see how long it takes for humans to understand the potential hazard of chemical. And there are other things, but as bisphenol A, phthalates. Phthalates, we had an episode, a plasticizer scandal in Taiwan. PBDE, triclosan, which many health effects are coming up. Of course, we shouldn't forget the pesticide that's very nicely reviewed by Brenda. It's an important event. And will it be banned? Probably never. So the problem is the studies, when you take effort to do the studies, you see more and more effects, and you know more how to prevent them. Talking about PCB, I happen to study a little bit on the PCB effects. This is the episode in 1979 affecting 2,000 people in Taiwan. And one of the effects we witnessed were the reduced proportion of male birth of Yuchen fathers. So those directly exposed have, those men directly exposed had fewer male offspring. And that percentage is about 45% boys and 55% girls for the 8 years after the exposure. And why is that so? We do not know, but such fact were also observed in the Seveso event, 1976. We try to find whether it could be the sex chromosome or other so-called aneuploidy, whether they are the causal factor. So we use fluorescence to show X chromosome, Y chromosome, and number 8. The only thing we found was that normal X over normal Y were a little higher in greater than 1.4. But I really cannot explain why this happened and whether it's really connected to what we found in this gender change in the second generation. Was unable to find aneuploidy or diploid. The other thing we were doing is looking at the methylation, DNA methylation in peripheral blood in children of exposed mother, I should say exposed woman, 20 years after the exposure. And we found that still a few things that are having methylation change of P greater than, I should say P lower than 10 to the minus 7. And most of them I have never heard of. Only I know about AHRR, which is aryl hydrocarbon receptor repressor. But interestingly, AHRR were much higher methylation in some regions and actually suppressed in methylation in other regions as comparing to control. If you look at the normal methylation in peripheral blood, you probably will find that standard error is usually very tight, probably 1 or 2%, very, very tight. Therefore, this is a huge difference. And these findings were also related to PCDF exposure at birth time or PCB exposure at birth time, and also associated with the children's IQ tests. And there are certainly many things that we do not know. Why these genes? Why increased or reduced what consequences will be there. So there are much knowledge gap which we need to work on. But not only that generation, Professor Skinner warned us that there are things like transgenerational or multiple generational inheritance. So it's both as male and female. Actually, the germline is in your body. So the next generation is affected. However, that's not enough. The F2 is again also affected. If an exposed person is a pregnant woman, then there is a F1, and the germline is F2 is already in the body. So he also showed evidence that the F3 could be affected by several of the environmental chemicals. And this is an important warning, but I truly do not know how we can do the study as an environmental epidemiologist. Previously, I know that as an epidemiologist, you have to live longer than your subjects or participants. Now you have to live longer than your participants' daughters or sons. So it's a big task. There are changes documented, methylation, non-coding RNA, histone modification, chromatin structure. One of the important outcomes in the children is the asthma prevalence, asthma. And this very nice review in 2005 found in many countries, if there are two studies, the two studies show almost ubiquitously increasing trend in asthma. Professor Marks told me that in Australia it's plateaued, but in many other countries, probably not yet. And as described by researchers, the atopic march actually is starting at 2-year-old. Followed, this is the eczema and food allergy, followed by asthma, and followed by allergic rhinitis. So we allow a study get carried out, especially at school age, but that's not sufficient because there are, if the outcome starts at 2-year-old, the exposure must be much earlier. So the environmental study has to be carried out much earlier. And here comes the birth cohort. The prenatal exposure and personal or hereditary factor, early life exposure are important. And it's also important to obtain biological samples for internal exposure or early effect or outcome occur early in life. We really envy our colleagues from Japan and Korea. They have, I saw our one of the director of the Korean birth cohort here. So there are plans to collect mother and child pairs for the next many years and to look at their health outcomes. I think it's just gorgeous and will provide much human knowledge. Taiwan has a birth cohort study that is bigger in size relatively big in size, collecting about 12% of all population, all births in 2005, and was regularly followed up. And we found from there, smoking in fathers were 50%, 60% of families were having incense burning, 40% of families use pesticides. And there are already findings, including exposure from indoor mold, maternal job stress, CO, ambient CO exposure, and the incense burning. But this cohort does not have biological sample. So actually missed a very important period of study. Why is biological samples important? For chemical exposure, biomarkers, I think this is a way to describe. There are exposure from air, food, water, and earth. And you can measure those all together in the internal exposure. You can measure early effect, preclinical conditions, disease. And it's essential that internal dose need to be corresponding to external dose. And it has to have some prediction of the disease. If any of the two conditions not here, that biomarker is not very nice, not totally nice. In children, additionally, exposure from transplacental or lactational also play important role. We try to compare human and the environmental marker, and I think human marker is probably more target for protection and with multi-purpose indicator and related to multiple exposure pathway. It has less relationship with the source. So we hope that development of regulation technology to early detect human exposure and early marker will be developed in a more applicable method. So we just mentioned about Taiwan's birth cohort study, which did not have biosample. Fortunately, we have three birth cohorts here, and there are also newer birth cohorts. And now an effort is trying to put the three cohorts together. These cohorts were organized by Professor Wang, Professor Paotong Chen, they are both in the room, and myself. I think a major task will be to harmonize the exposure assessment and harmonize their outcomes, and still need to overcome the small number of participants. So we already know that many health effects been documented in children from the environment. So the third question is it possible to prevent environmental events? In 2011, one lab staff of Taiwan Food and Drug Administration, the Ministry of Health and Welfare, detected DEHP level of about 600 ppm in probiotic supplements. And then investigation was started, and then MOHW announced that there are already perfume or cosmetics that are exposed, were contaminated. And many foods were found to be contaminated as well. So they need to notify the International Food Safety Authority Network, or WHO, and the Rapid Alert System of Food and Feed of the European. These are, and so many products from Taiwan cannot be exported. And there are so many things that's found to be contaminated. So at the end, in the end of year 2011, it was estimated a financial loss only in half year, $500 million in Taiwan. So I don't know how much after 2011. I think no one dare to estimate and cannot be accurately estimated. So after that, the National Health Research Institute received a grant. That's a big orthodox, I should say. Very strange. So when there is an episode, the environment finally got noticed that you are important. So that's how these studies are done. But it was found that actually exposure at certain level were associated with health effect in the children, including sex hormone binding, globulin, reduced height and weight, and IGF-1, and microalbuminuria, and increased asthma. So, well, it's good to have study findings, but I think the most important question is, can similar effects as the plasticizer scandal be prevented in the future? I think it's an important question for all people here. Happened that I participated in one study looking at serum and spot urine sample collected from 72 Taiwanese pregnant women. No, I didn't participate. I was one of the researchers, sorry. And the thyroid function and sexual hormones were studied, and salates metabolites were measured. At that time, we are excited to find that there was relationship between the urine MBP, which is a metabolite of DBP, a negative relationship with free T4. The first author, Dr. Huang, is also here. There, yeah. However, one important thing we kind of miss. The pregnant women were 4.6 times higher than US pregnant women in MBP and 13 times higher in MEHP. Can you believe we didn't notice that? Why were we not alarmed that something wrong was already and actively happening that time? And I think that's not allowable for the future. So I am trying to connect with this Nashid study. You have to be careful about the pronunciation. But to monitor the nutrition status, health status, they do lifestyle and health and nutrition related knowledge and attitude practice in the whole population representative samples. And that are representative for age, sex, geographic, and ethnic group. And they keep on rolling and collected 1,000 to 2,000 samples per year. And I think it's useful if we can get connected with them. Is it possible to prevent future environmental events by adding environmental biomarker to this? And if that can be done, what we should do? I believe we should do more continuous recruitment. We need a little higher number of participants and probably a higher, And the measurement of our potential contaminants from our toxicologists. And early determination for analysis of suspected chemical contaminants. And also an algorithm to show us when to alarm. And I think this is what we lacked back in 2007. This is the final slide. I want to show Taipei. This is a famous or infamous Taipei Bridge, very crowded with many motorcycles. So some people are brave enough, and I'm sure he will be successful with such courage. And I am not sure they will do this in the future. With that, I will thank you very much, and please visit us at NIEHS Taiwan. Thank you.